A nociceptor is a sensory nerve cell that responds to damaging or potentially damaging stimuli by sending signals to the spinal cord and brain. This process, called nociception, usually causes the physical sensation of pain in sentient beings. History. Nociceptors were discovered by Charles Scott Sherrington in 1906. In earlier centuries, scientists believed that animals were like mechanical devices that transformed the energy of sensory stimuli into motor responses. Sherrington used many different styles of experiments to demonstrate that different types of stimulation to a nerve's receptive field led to different responses. Some intense stimuli trigger reflex withdrawal, autonomic responses and pain. The specific receptors for these intense stimuli were called nociceptors. Location. In mammals, nociceptors are sensory neurons that are found in any area of the body that can sense noxious stimuli either externally or internally. External examples are in tissues such as skin, cornea and mucosa. Internal nociceptors are in a variety of organs, such as the muscle, joint, bladder, gut and continuing along the digestive tract. The cell bodies of these neurons are located in either the dorsal root ganglia or the trigeminal ganglia. The trigeminal ganglia are specialized nerves for the face, whereas the dorsal root ganglia associate with the rest of the body. The axons extend into the peripheral nervous system and terminate in branches to form receptive fields. Development. Nociceptors develop from neural crest stem cells. The neural crest is responsible for a large part of early development in vertebrates. More specifically, it is responsible for development of the peripheral nervous system. The neural crest stem cells split off from the neural tube as it closes, and nociceptors grow from the dorsal part of this neural crest tissue. They form late during neurogenesis. Earlier forming cells from this region can become non-pain sensing receptors, either proprioceptors or low-threshold mechanoreceptors. All neurons derived from neural crest, including embryonic nociceptors, express the TRKA, which is a receptor to nerve growth factor. However, transcription factors that determine the type of nociceptor remain unclear. Following sensory neurogenesis, differentiation occurs and two different types of nociceptors are formed. They are classified as either peptidergic and non-peptidergic nociceptors. The sets of receptors express distinct repertoires of ion channels and receptors. With the specialization, it allows the receptors to innervate different peripheral and central targets. This differentiation occurs in both perinatal and postnatal periods. The non-peptidergic nociceptors switch off the TRKA and begin expressing RET. RET is a transmembrane signaling component which allows for the expression of another growth factor, glial cell-derived growth factor. This transition is assisted by RUNX1, which has proven to be vital in the development of non-peptidergic nociceptors. On the contrary, the peptidergic nociceptors continue to use TRKA and they express a completely different type of growth factor. Currently there is a lot of research being done to determine more specifically what creates the differences between nociceptors, types and functions. The peripheral terminal of the mature nociceptor is where the noxious stimuli are detected and transduced into electrical energy. When the electrical energy reaches a threshold value, an action potential is induced and driven towards the central nervous system. This leads to the train of events that allows for the conscious awareness of pain. The sensory specificity of nociceptors is established by the high threshold only to particular features of stimuli. Only when the high threshold has been reached by either chemical, thermal, or mechanical environments of the nociceptors triggered. The majority of nociceptors are classified by which of the the environmental modalities they respond to. Some nociceptors respond to more than one of these modalities and are consequently designated polymodal. Other nociceptors respond to none of these modalities and are referred to as sleeping or silent. Nociceptors have two different types of axons. The first are the A-delta fiber axons. They are myelinated and can allow an action potential to travel at a rate of about 20 meters per second towards the CNS. The other type is the more slowly conducting C-fiber axons. These only conduct at speeds of around 2 meters per second. This is due to the lighter non-myelination of the axon. As a result, pain comes in two phases. The first phase is mediated by the fast-conducting A-delta fibers and the second part due to C fibers. The pain associated with the A-delta fibers can be associated to an initial extremely sharp pain. The second phase is a more prolonged and slightly less intense feeling of pain as a result of the acute damage. If there is massive or prolonged input to a C fiber, there is a progressive build-up in the spinal cord dorsal horn. This phenomenon is similar to tetanus in muscles but is called wind-up. If wind-up occurs, there is a probability of increased sensitivity to pain. Thermal nociceptors are activated by noxious heat or cold at various temperatures. There are specific nociceptor transducers that are responsible for how and if the specific nerve ending responds to the thermal stimulus. The first to be discovered was TRPV1, and it has a threshold that coincides with the heat pain temperature of 42 degrees Celsius. Other temperature in the warm-hot range is mediated by more than one TRP channel. Each of these channels express a particular C-terminal domain that corresponds to the warm-hot sensitivity. The interactions between all these channels and how the temperature level is determined to be above the pain threshold are unknown at this time. The cool stimuli is sensed by TRPM8 channels. Its C-terminal domain differs from the heat-sensitive TRPs. Although this channel corresponds to cool stimuli, it is still unknown whether it also contributes in the detection of intense cold. An interesting finding related to cold stimuli is that tactile sensibility and motor function deteriorate while pain perception persists. Mechanical nociceptors respond to excess pressure and mechanical deformation. They also respond to incisions that break the skin surface. The reaction to the stimulus is processed as pain by the cortex, just like chemical and thermal responses. These mechanical nociceptors frequently have polymodal characteristics, so it is possible that some of the transducers for thermal stimuli are the same for mechanical stimuli. The same is true for chemical stimuli, since TRPA1 appears to detect both mechanical and chemical changes. Chemical nociceptors have TRP channels that respond to a wide variety of spices. The one that sees the most response and is very widely tested is capsaicin. Other chemical stimulants are environmental irritants like acrolein, a World War I chemical weapon and a component of cigarette smoke. Apart from these external stimulants, chemical nociceptors have the capacity to detect endogenous ligands and certain fatty acid amines that arise from changes in internal tissues. Like in thermal nociceptors, TRPV1 can detect chemicals like capsaicin and spider toxins. Sleeping, silent. Although each nociceptor can have a variety of possible threshold levels, some do not respond at all to chemical, thermal or mechanical stimuli unless injury actually has occurred. These are typically referred to as silent or sleeping nociceptors since the response comes only on the onset of inflammation to the surrounding tissue. Polymodal. Many neurons perform only a single function, therefore neurons that perform these functions in combination are given the classification polymodal pathway. Afferent nociceptive fibers travel back to the spinal cord where they form synapses in its dorsal horn. This nociceptive fiber is a first-order neuron. The cells in the dorsal horn are divided into physiologically distinct layers called laminae. Different fiber types form synapses in different layers, and use either glutamate, a substance P as the neurotransmitter, or delta fibers form synapses in lamina A and V. C fibers connect with neurons in lamina 2A. Beta fibers connect with lamina I3 and AMP-V. After reaching the specific lamina within the spinal cord, the first-order nociceptive project to second-order neurons that cross the midline at the anterior white commissure. The second-order neurons then send their information via two pathways to the thalamus, the dorsal column medial lemniscal system and the anterolateral system. The first is reserved more for regular non-painful sensation, while the lateral is reserved for pain sensation. Upon reaching the thalamus, the information is processed in the ventral posterior nucleus and sent to the cerebral cortex in the brain via fibers in the posterior limb of the internal capsule. As there is an ascending pathway to the brain that initiates the conscious realization of pain, there also is a descending pathway which modulates pain sensation. The brain can request the release of specific hormones or chemicals that can have analgesic effects which can reduce or inhibit pain sensation. The area of the brain that stimulates the release of these hormones is the hypothalamus. This effective descending inhibition can be shown by electrically stimulating the periaqueductal gray area of the midbrain. The periaqueductal gray in turn projects to other areas involved in pain regulation, such as the nucleus raphe magnus. In turn, the nucleus raphe magnus projects to the substantia gelatinosa region of the dorsal horn and mediates the sensation of spinothalamic inputs. The periaqueductal gray also contains opioid receptors, which explains one of the mechanisms by which opioids such as morphine and diacetylmorphine exhibit an analgesic effect. Sensitivity. Nociceptor neuron sensitivity is modulated by a large variety of mediators in the extracellular space. Peripheral sensitization represents a form of functional plasticity of the nociceptor. The nociceptor can change from being simply a noxious stimulus detector to a detector of non-noxious stimuli. The result is that low-intensity stimuli from regular activity initiates a painful sensation. This is commonly known as hyperalgesia. Inflammation is one common cause that results in the sensitization of nociceptors. Normally, hyperalgesia ceases when inflammation goes down. However, sometimes genetic defects and/or repeated injury can result in allodynia. A completely non-noxious stimulus like light touch causes extreme pain. Allodynia can also be caused when a nociceptor is damaged in the peripheral nerves. This can result in deafferentation, which means the development of different central processes from the surviving afferent nerve. With this situation, surviving dorsal root axons of the nociceptors can make contact with the spinal cord, thus changing the normal input. Nociceptors in non-mammalian animals. Nociception has been documented in non-mammalian animals, including fish and a wide range of invertebrates, including leeches, nematode worms, sea slugs, and fruit flies. Although these neurons may have different pathways and relationships to the central nervous system than mammalian nociceptors, nociceptive neurons in non-mammals often fire in response to similar stimuli as mammals, such as high temperature, low pH, capsaicin, and tissue damage. Terminology. Due to historical understandings of pain, nociceptors are also called pain receptors. This usage is not consistent with the modern definition of pain as a subjective experience.